All right.
So, hey, Chris. Oh, sorry. Is screen sharing enabled right now?
I need to do that. Cool. You are now a co-host. So, should be able to share your screen. Perfect. So yeah, I'm very excited to introduce Sebastian Swanson. Sebastian's a graduate student in Amy Keating's lab at MIT, and he's going to be sharing with us the story of his thesis work so far. And yeah, Sebastian, please take it away.
Cool. Thanks, Chris. Yeah, I never really thought that the first talk I gave in grad school on my research would be over Zoom, but here we are. Okay, so today I'm going to talk about data-driven design of custom-fit protein-binding peptides, and I'll walk through all those hyphens and kind of describe what I mean over the next few slides. But I'm Sebastian. I'm in Amy Keating's lab in MIT Biology. I'm a fourth-year grad student, and this is very much like a work in progress. So we're really excited to have you guys in particular as an audience to kind of hear what we're working on and provide some feedback and give us your thoughts. So this should be pretty fun. Okay, no further ado. Okay, so I think that with this audience, I don't really have to convince you guys that protein binding molecules are incredibly useful. I think you're aware of the different applications. In general, you can think of them as useful for detection, so binding some kind of protein in a complex mixture. They're useful for inhibition of interactions between proteins, and they're also useful for activation. You could either bind something and stabilize some conformation that has enzymatic activity, or you could stabilize a dimer. There's all kinds of applications for these. While it's currently difficult to generate binders, the ability to do this on demand would have big implications for science and medicine as a whole. So today I'll talk about the method that we're working on in our lab to design these protein binding peptides that can potentially fill this role. So the Keating lab has a long history of studying and designing peptide molecules, so you could argue that we're a little biased. But in general, I think that peptides have a lot of benefits as binding molecules. In comparison to small molecules, peptides are genetically encodable. This means they can be developed and refined by directed evolution, and also that anything with ribosomes can be used to produce them. They're also better suited for binding protein surfaces, mostly because those are quite large and flat, and it's kind of difficult for small molecules to wedge their way in there and bind selectively. In comparison to antibodies, peptides are more stable. So they're smaller and lack post-translational modifications. This means they can function in the cytosol and they're easier to express. You don't need complicated mammalian cell lines. So while they have nice properties, it's of course not so trivial to actually obtain new protein binding peptides. So today I'm focusing on a computational method for doing this. So I'm going to talk about de novo design in kind of the general manner. So generally, so far this problem has kind of been approached as a two-step process. The first step is designing a backbone, and then the second step is designing a sequence that will adopt that backbone conformation. In case that we want to design a backbone that binds a protein, you just modify this and you prioritize favorable interactions between the peptide and the protein at the step of sequence design normally. I imagine we're all pretty familiar with sequence design. Here I'm kind of just showing an example of some intended conformation. We're thinking of designing this green curly bit. You can kind of imagine for all the possible sequences that each has this kind of conformational energy landscape. And the sequence that you want is the one that has a free energy minima that corresponds to your desired conformation. Obviously with design, we don't actually search this whole energy landscape, but that's kind of what a successful design looks like in terms of designing sequence. Backbone design, in particular for non-idealized proteins that take on specific functions, I would argue is less explored. We can limit the space of potential backbones to those that exhibit recurring structural patterns or fragments from known proteins. But even if we do this, there's still no guarantee that if you make a backbone out of these fragments, that there is some sequence that will take on this desired conformation. So here I'm just showing you another squiggly, but this one just doesn't happen to have any sequences that will fold into that structure. So I'm going to use some jargon, and I think this is probably what I'll use the most, but this idea of designability versus non-designability. You would say that this first thing is designable and the second thing isn't. So I thought it would be interesting to just kind of look at an example of kind of a state-of-the-art application of our methods that we have today for designing protein binders. So I have this example from the Baker lab where they very recently designed tight mini protein inhibitors of the SARS-2 spike protein. And overall, they kind of treated this like a 3-step process. The first step was to generate these backbones, like mini protein backbones. They also refer to them as scaffolds. Then they docked these to the spike protein at this binding site that they wanted binders to interact with to act as inhibitors. And lastly, they designed sequence for the new interface. So on one hand, I think since the goal was just to obtain a tight binder, this is definitely a success. But on the other hand, I think that if less designs had been tested, it's possible that this method wouldn't have yielded a hit. So I think this success rate reflects kind of the difficulty of the problem and also the potential for improvement. One thing that I think is interesting is the fact that all mini protein backbones were generated 3 years ago in a separate paper in a manner that prioritized their designability as opposed to their ability to interact with the binding site. So there's obviously different reasons why this might have not been as successful as it could have been. But I'm kind of pinpointing on this because this is something that I think my method kind of addresses. So to give some intuition for why there may be limitations to designing a backbone independent of the site you would like the mini protein to interact with, I wanted to use some classic lock key cartoons. These are kind of inspired by something like a talk I saw Chris gave a while back. So we can think of the key as like a binder, and it interacts with our target, the lock. And just to make this analogy paper thin, I'm differentiating between the colored bits, which I'm thinking of as side chains, and the actual backbone, which is this gray base. And the backbone acts as a scaffold, and it presents the side chains in a specific way, specific orientation, and places them such that they can interact with the lock. So to successfully interact, the side chains have to be paired with their partners. And there basically need to be no spaces in my analogy. So this is an example of a good interaction, a true key lock pair. So you can kind of think about docking scaffolds or backbones as taking a set of pre-generated key scaffolds and just kind of asking whether or not they fit. This is kind of an analogy to the Rotamer interaction field method, which I didn't really describe, but some of you may be familiar with. I'm showing you this lock plus the desired interactions. There's the side chain that would be actually part of the protein or the lock. And then there's also the side chain that would interact with it that ideally our key should be able to sustain. So we can try our library of backbones, and if one fits and supports these interactions, then it's great. We have something that works. But if it so happens that one doesn't, or that it's difficult to find things that are complementary, then we're kind of out of luck. So I think another approach, an alternative, would be to actually just build the backbone to fit the target. And so just to kind of illustrate what I mean, I'm kind of showing this example again of this target lock that we want to design an interaction to, and sort of these examples of key lock pairs that we find out in the wild. And I'm going to kind of argue that we can kind of learn from these. So in particular, we can look for regions of local similarity. So here I'm just drawing boxes between similar regions of the target lock and these known key lock pairs. And if we have enough of these, we can kind of describe all of the target lock binding site with examples from nature. So we can extract those bits that correspond to the key that interacts with a part of the binding site that we want to engage. And we can kind of merge them together to make some kind of Franken key type situation that takes on all these properties. And then, as you might expect, if you fit the proper side chains onto this, it can dock the target lock successfully. So that's sort of the premise for what I want to do. Obviously, there's caveats. For example, we're prioritizing the interaction as opposed to the stability of the lock itself. But I'll kind of explore that as I talk more about this method. So obviously, we're not trying to build keys. We want to design peptides. So if we want to do that, we need to use the appropriate building blocks. And to do that, I'm kind of thinking of, revealed one thing too many, using 2 segment fragments. So these are these are fragments from protein structures. You can think of from peptide protein complexes, taking parts of the peptide and interact with the protein, and then kind of using those lock key pairs to design new peptides. And if we had an exhaustive database of peptide protein structures, this would work great, but we don't. So we can't really use those as our source of building blocks. Another way to do this, though, is to use information or structural data from protein cores, which are much better represented in the PDB. The Gregorian lab and Gevorg has done a lot of work exploring structural motifs that encapsulate tertiary interactions. These tertiary motifs describe interactions that are distant in sequence but close in space. And like when I say tertiary or terms, I'm referring to tertiary motifs. So here they just kind of identified the most common tertiary motifs in the PDB, which, you know, if you guys look at a lot of proteins, they probably look really familiar to you, like beta strands next to one another, alpha helices interacting, that kind of thing. Surprisingly, only 24 of these are necessary to cover a third of the structural elements in the PDB. So they're highly recurring, and they're potentially also useful for designing peptide protein interfaces. That's kind of what we hypothesized with this project. So we devised a method to kind of use terms to discover potential like new peptide bonding conformations. So this kind of starts by identifying the region that you might want to design a peptide to interact with. Here I'm showing white space. This like white sphere is representative of a residue on the protein that's like in this binding site that we want the peptide to interact with. We can define a fragment around this residue that kind of captures like a meaningful amount of structure of the binding site. And then we can search for matches to that fragment in other proteins. So here you can see that like that original fragment, there's a very similar example, or something very similar to it in the middle panel, but also this like new element that co-occurs with it on the left. And that like new bit, that new like strand of like structure is a good candidate for something that we can bring back into the space around the target protein and potentially use for designing new backbones. So I'm going to refer to this as a seed just because it represents like a building block that can combine with others to grow a peptide. So here's this render. I had a really nice undergrad who helped me develop a way to like visualize this. And here the like lines represent individual seeds. And like at first glance, it just looks like a mess. We would affectionately refer to it as the hairball. But like each of these hairs represents like a seed. And if you look closely, you start to see like patterns in the way they're arranged. It's not just random. The colors represent different secondary structures. And I think the first thing we noticed really looking at this is that the beta strands in particular tend to kind of cluster around unpaired beta sheets. So I'm circling one of those examples here. And there's also differences in density overall, where there's just more seeds in some areas. So we kind of like, we saw this and we knew that there was like information to be gleaned and that this could potentially be useful, but we kind of needed a method to actually translate that into peptide backbones. So to make these useful for design, we developed this method for combining seeds. And the basic idea was to search for overlaps within the cloud of seeds. So here the red boxes just represent like close overlaps. Like realistically, we'd want them to overlap more closely than this. But the idea is that these are potential junctions. So seeds can be combined at these points into something larger that could potentially be used as a peptide backbone. Okay, so that's the goal. But before we got to that, we took a step back and asked like whether these hairballs even contained seeds that were similar to peptides. Because if, you know, we would like to see that we could design native peptides or peptides that we know exist before we try to go design new things. So we kind of framed this as a coverage benchmark. And our goal was to find seeds that cover peptides, at least like at a small local level. So just to walk through this, I know there's a lot of text. We start on the left. We take like a real peptide protein complex and define a binding site on the protein. Those are just residues that can interact with the peptide. And then we generate seeds using the method I described, and then ask whether in that big space of seeds, there are some that overlap really closely to the peptide. And that's based on this RMSD criteria that looks at like the actual backbone atom overlap. And then we can kind of like add this up and ask like how many overall of the peptide residues are described by some seed. So in this example, the end residues on the C and N terminus like aren't covered. So, but the rest of them are. So we'd say like 7 out of 9 peptide residues are covered in this case. In terms of like the actual definition of like overlap or alignment, so this for defining coverage, you have to say whether something is the same or not. So we use this definition of RMSD, and we had we defined a cutoff of 5 residues with the cutoff being 1.25 angstroms. And this is like, it might seem a little bit arbitrary, but we kind of explored cutoffs different RMSD like thresholds and also with different lengths. And we kind of found that for longer lengths, it was just kind of unlikely to find overlaps to the peptide. And for shorter lengths, they weren't really useful for this idea of like combining seeds together because we needed overlaps between those seeds. So anyways, this length ended up being sort of a sweet spot. So to do this analysis kind of at the largest scale possible, we opted to use PixelDB, and it's a database that our lab compiled of like high quality peptide protein structures. And on the left, I'm just showing some examples. So overall in this like database, there's almost 2,000 structures, but if you like group the binding sites into like classes by looking for structural similarity, you find that there's only about 486 or exactly 486 binding sites classes. So we took a representative from each class and generated seeds in that binding site. And here I'm just showing you like that there's kind of a wide range of peptide sizes in this database. And for some of the peptides, they're actually, you know, a lot more like mini proteins, and not all of the peptides were part of the interface. So we kind of restricted this question of coverage to interface residues, which we defined as residues that have at least one contact to the protein, and also that have a B factor, which is not so high relative to the like average B factor for that complex. So at first glance, the result was just that peptide interfaces under this definition of coverage are largely covered by seeds. So overall, 83% of the interface residues in the dataset were covered. And if we broke that up by individual peptides, we found that more than half of the peptides were 100% covered. And those would look a little bit like this. So in the red, I'm showing the peptide, in the blue, I'm showing like the seeds. And they overlap quite nicely, and like all of the interface residues are covered. But there are also examples in the middle where like not all of the residues were covered. So here I'm showing you, this case where only half are covered. And also at some of the regions that are covered, the overlap isn't quite so nice. And then finally, there's this third class of peptides that were not covered whatsoever. So we wanted to understand like why like 15% of these peptides were like completely like uncoverable by seeds. So to answer this question of which types of peptides are not covered, I kind of divided the peptides based on whether they were covered or not, and then like looked first at their length. So here I'm just showing you like a histogram describing the length of the covered versus not covered peptides. And the conclusion, as you might expect, is that the peptides that were not covered by seeds were much shorter than the rest. This makes sense in kind of like a trivial sense because there's less positions. So it's like more likely that you might happen across a peptide that you can't cover at all. But also it reflects, I think, that this is a really small percentage of the data set. Like, so we're covering like the vast majority of like actual interface positions. We also found that the secondary structure of the peptides that weren't covered was predominantly random coil and turns. This isn't super surprising since the database that we're generating the seeds from has roughly the same secondary structure distribution as the PDB, which is predominantly helix and beta strands. But it doesn't mean that we're not unable to cover random coil and turns. So there's a lot of examples of those that are covered in this data set. While it was really promising to see that seeds could, for the most part, cover native peptide structures, we began to wonder if this was like too easy of a test. So before, the render I showed you was like considerably subsampled. I was showing you 5,000 seeds out of like 2 million. And if you kind of like imagine how dense the space is, it's kind of easy to think about just like them randomly overlapping a peptide. So like this question is kind of like, if we flood the binding site with tons and tons of fragments of protein structure, is it just the case that we're going to end up getting coverage? So to kind of formalize this idea of flooding the binding site with seeds, we kind of framed it, so we wanted to formalize this idea and see if that was sufficient, flooding the binding site with fragments of protein structure to cover peptide residues. So to see if that was sufficient, we framed it as kind of a problem of disproving this null hypothesis. So the null hypothesis is basically that seeds are random bits of protein structure arranged around a binding site. And we don't think that's true. We think our seeds are obviously like positioned in ways that are based on like protein, like tertiary motifs. So they should be better than random, but this null model should reflect what like the random case is. So to generate a cloud of seeds that was essentially random, I essentially shuffled each term seed distribution. So I did this by taking each term seed and then replacing it with a new fragment of the similar length, same length, with a new conformation. and then sampling like a new pose within like some range that I had defined as the binding site. So this held all the variables constant, like number of seeds and their lengths and like the range over which they are distributed, but like randomized their conformation and pose. So I'll refer to these as null model seeds. And the next question was, of course, like whether or not these could cover the peptide. So here I'm just showing you the plot I showed before of like what percentage of peptides are covered, and then also that for null model seeds. So on average, like you would expect, most of these peptides are not covered by null model seeds. But interestingly, like there were a lot of peptides that were either partially and some that were even completely covered by these null model seeds, which is not exactly what we'd expect if this was really a hard problem. So in one sense, this isn't like a bad thing. It just means that we can recapitulate those binding modes randomly. But in another sense, we really wanted to like see whether or not our like seeds were reflecting like preference for actual binding modes and not just like every possible way to arrange a fragment in the binding site. So we became interested in ways to like filter seeds, and I'll try to walk through this. I'm sorry, it's like a lot of images and stuff. But the first way we tried to filter seeds was by ranking the structural matches to the binding site fragments by RMSD. So before I was talking about how we define a fragment and search against the database, and we get like, in this case, I was using up to 100,000 matches to each of those fragments. I can rank them by RMSD and then say, okay, if I only look at the top best 10,000, like how good is the coverage? If I only generate seeds from those top matches, how good is my coverage? So I varied from 5,000 to 100,000 matches. And that's what I'm showing you on the top left. And the basic trend was that coverage increased as we used more matches to generate more seeds, which is exactly what you expect. But interestingly, we found that the rate was very different between the term and null model seeds. So the term seeds like increased really quickly. You only need 10,000 matches per binding site fragment to cover most of the positions. And then after that, it increases at a rate that's almost similar to the null model seeds. So this is really curious, and it seemed to suggest something special about the first 10,000 matches. So I tried rearranging the order, and instead of like sorting by RMSD to the binding site, I just randomly shuffled the order, and I also tried reversing it. And I found that, like, as you would expect, these decreased the rate at which coverage increased at first, because we weren't using like the best possible matches, but it didn't really change it by that much. So it didn't seem like there was something special about the top 10,000 matches. It just seemed like overall seeds were kind of like rediscovering similar kinds of binding modes. And you could do that with like really low RMSD matches and with higher RMSD matches as well. Going forward, actually, for like other applications, I was able to use this to like justify using many less seeds, which made it a lot easier to do some of these like downstream things. Okay, so now I'm going to kind of change gears and just talk about the problem of actually applying these to designing peptides. And we've been working on developing some tools to do this, but we're still kind of just doing small scale tests to understand how they work and how to like refine them. So that's kind of the goal of this. I'm not trying to prove to you that I've like designed a bunch of like peptides that will bind if we go test them. Since our lab studies peptide interactions with the EVH1 domain, it made sense for me to consider this as a potential target for de novo designed binders. Recently, we characterized a binder of this domain, which is top secret, so I can't show you, but it binds in this binding site, which you can kind of see defined by these like black spheres. Those are the binding site residues. And in the middle, you can see the fragments defined around those binding site residues. So that just gives you kind of an extent of like the range of the protein that I defined as the binding site. And then actually this image I've been showing you before of like seeds was for this protein that I want to generate binders to. And you can see that the like space of seeds that's occupied is even wider than the space of the binding site. So there's quite a lot of range of like areas in which you might expect things to bind the protein, or at least are designs to interact with the protein, not necessarily bind. Okay, so like once you generate the space of seeds, you have to like, the next step is to identify overlaps and represent them in a way that allows us to sample new backbones. So we chose to represent the seeds as a graph where the nodes represent residues and the edges represent either bonds observed in seeds or potential bonds that you can make between seeds suggested by overlaps. This is, I think you guys might be able to tell, but this is very much inspired by sowing. which uses kind of a similar approach to sample paths from a graph and generate backbones. But anyways, since the overlaps needed to be similar enough to be joined together, we opted for a rather strict definition of like overlaps. We defined them over 4 residues. And we had the requirement that like no pair of overlapping atoms from the 2 seeds was more than 1 angstrom apart. That's slightly different than like the RMSD definition that I showed you before for coverage. And we actually just found that this was faster to compute. And actually, this was not sufficient in some cases to ensure that the seeds overlap nicely. Sometimes we get seeds that were similar but had kind of different orientations. So we added this extra criteria that the angle, which you can define from like, basically you define like a basis vector for each residue, that on average the angle deviates no more than 45 degrees. So we use this definition to define overlaps, and in the graph, I'm kind of just showing you with the diagonal dotted lines, like the potential bonds, which we define based on overlaps. And kind of as a sanity check, I checked at how many overlaps we observe when we try to find them for term seeds versus null model seeds. And like you might expect, since the null model seeds are arranged randomly, we would expect rather that there are less overlaps, and that's consistent with what we're seeing. But I was actually pretty surprised by like how drastic this difference is. So that was promising. That indicated that the term seeds were arranged in a way that was like special for not just covering the peptide, but also maybe being combined with one another. So next we sample the graph, and I'll just show you like how we do this. To do this like uniformly, or not necessarily uniformly, that'd be a little bit more complicated, but just to sample a random path, you can pick a residue from the graph and then extend it. If there's a point where there's multiple outgoing edges, you can just pick one randomly and then continue until you reach like a C terminus. You can repeat the same thing until you reach the N terminus, and that gives you like a path. So this is like what a path is, really just a set of residues that we've selected from these different seeds, and also like how they correspond to one another. So like which one comes first, second, etc. So that's kind of like the information contained in that. But the next step was just to ask whether we would accept this. We had some really basic criteria, like the path should be unique, not yet sampled. It should be above some minimum length. You can imagine how you can sample paths that just span 1 seed, which isn't really interesting. And also we would, after passing these criteria, I would fuse the paths. So that's just like this program developed by the Gregorian lab, which they use to kind of take a set of overlapping structures and merge them into like a consensus structure. There's lots of different ways to do this, but this is the one I used. And if the resulting backbone didn't have any clashes, then I would accept it. So I sampled 4,000 backbones, but then my first question was, of course, like, what is the diversity of these 4,000? So I just computed like RMSD over like their backbones, and then clustered based on this. So on the left in this like really busy plot, I'm just showing you the result of like hierarchical clustering of all of the 4,000 backbones. So this allows you to kind of define clusters at different RMSD cutoffs. And of course, as the RMSD cutoff increases, you get less clusters because the clusters increase in size. To like get a sense of overall diversity, I first tried setting the RMSD high such that I got 16 clusters. And here are just like representatives from each of the 16 clusters. And the first thing you might notice is that these are like mostly just helices, but there is variation in like how they engage the binding site and also like their own structure. Sometimes there's like kinks in the helices. So it's not as simple as just kind of like copy pasting idealized helices like in the binding site. And there are also some like extensions outside of the helices that had like non-helical structure. So these were kind of interesting, and they represented at least a range of ways to engage the binding site. But going forward, I thought that what I wanted to do at this step was really just like eliminate redundant structures. So I chose a really tight cluster cutoff, just 1 angstrom. And I considered anything closer than this to another thing like redundant, and took one example from each cluster. So that was 1,764 peptide backbones. So I have this whole set of peptide backbones, and they're potentially like designable. Potentially you can design a sequence and they'll bind, but it's kind of expensive to figure that out by just like designing sequence. So we're interested in ways to kind of filter those backbones down and prioritize the ones that we think were like designable or likely to bind. I devised these kind of like different filters, and I arranged them from like from fastest to compute to slowest to compute, so that we could like waste less time computing really slow things for peptides that we're not going to be interested in. So I'll walk through these. The first step is just looking at contacts. This is like a sequence independent definition of contacts, which I won't go into, but the basic idea is that two residues are contacting if they're positioned in a way such that they can, such that if you like put rotamers onto them, they're likely to interact. So it's like kind of potential to contact. So based on this, I was able to just look at how many contacts each peptide would make with the protein and kind of like weed out the ones that made very few contacts. There were some that were like oddly distant from the protein.
Sorry, Sebastian, if I ask a quick question on this, is really neat. So is this this contacts definition, is this code that you guys developed, or is this sounds also like the motif score in Rosetta?
Sorry, yeah, this is a Gregorian lab thing, which like a lot of these are Gregorian lab tools that I've like kind of repurposed in my time. But yeah, I guess, are you saying there's another application in Rosetta that does something similar?
Yeah.
Okay, cool.
I was just curious what the tool was, but yeah, that's neat.
Okay, yeah, so the basic idea was just to prioritize things that actually make interactions or can make interactions with the protein. The next step was to ask whether or not, like, if we define interface fragments around these contacts, whether they're like designable. And we defined designability in this case as just like whether or not we could find a match to this interface fragment in the structured database. You might think this seems a little redundant just because like we generated the seeds already using terms, but like the seeds were generated only in the context of part of the binding site, not all of it. So when you place them in that new space, they might interact with new things, and they might not be compatible with the other elements of the binding site. So this is kind of an important check to see if we've put a seed in a place that it won't be able to exist in. So if the peptide had a non-designable interface contact, we took this as evidence that it might not be designable overall. Obviously, that doesn't necessarily mean it's not designable. It just means we haven't observed anything that's quite like it. But just given how, like at this level of structure, most of like the PDB is pretty saturated of tertiary motifs, we think it's a pretty safe bet that this might not be designable. So previously I talked about designability in kind of like a binary sense, like yes or no. We can also quantify it in a way that allows us to rank peptides. So we can take two interface fragments and ask which is more designable, as in which is like, which fragment is able to be achieved by more like sequences and more contexts. So basically what's more common. And a simple approach to this is just to count the number of matches to a fragment in the database. So you can do this and you can compare fragments. And also, since our protein has a fixed sequence, you can also constrain your search based on the sequence of the protein. And this is kind of just saying like we want to count how many fragments exist given this specific sequence. So I used kind of all these metrics in these three different steps. And here I'm showing you the results, which I know is a lot, but we'll start from step one. So I computed the contacts and I looked at the average number of interface contacts per peptide residue. And there was kind of a wide range of like amount of contacts. I also computed the number of contacts between peptide residues. And there's also a wide range of number of contacts, but like not so much. It was really centered on like two contacts. And I think that's because of all the helices, which have the like I+4 contacts on both sides. But I didn't really know exactly like what the right number of contacts is on average. There's not really an answer for that. One way to do that, to get some insight into that, would be to look at the distribution for the native peptides. But in this case, I just kind of took the top 50th percentile, as in like these are better than the other ones. And I did that for both the interface contacts and the intrapeptide contacts. I then looked at the designability of the interface in the binary sense, and looked at that for both the interface contacts and the intrapeptide contacts. And kind of surprisingly, I found that for the most part, these were entirely, like every interface fragment and every intrapeptide fragment was designable. So in some sense, it was promising because like that indicated that like we didn't have really terrible designs, but at the same time, it means that this isn't really a useful metric for filtering things. So here I just said if anything had a fragment that wasn't designable, that I would like not continue with it. So I filtered those out. And then finally, I picked like 15 peptides to score with like this more complex, like actual rank kind of approach that I was talking about before. The specific score I computed here is different than what I described in the last slide, but it still fundamentally measures like sequence structure compatibility. And here I'm just showing you that like the experimentally verified peptide ranks pretty high. It's second to highest out of all of these. There is one thing that ranks a little higher. It's actually kind of unfair because the thing that ranks better than this it has like one piece that interacts with the protein a lot and one piece that doesn't. So it's like it would be more impressive for something to have a really good sequence score and also make a lot of interactions, like more interactions than the native peptide with the protein, but that's not the case. But anyways, since there were designs that were kind of in the range of the native peptide, I decided to take like the top 5 of these and do sequence design. I probably won't be able to satisfyingly explain this here, but to design sequence, I opted to use Determine, which was also developed by the Gregorian lab. You guys may be sensing a trend. This method uses sequence structure relationships in the PDB to estimate how suitable a given sequence is for a protein backbone. It's kind of like, I think of it as the probability of a sequence given a structure. And it does this by building a sequence level model, which captures the sequence preference of each position in the protein independently. And also it captures them in kind of like a pairwise sense, looking at interactions between positions. So to actually like do this, to get like both the self and pair energies, you have to search basically parts of the protein, like fragments, and then count like within that fragment how often you occur each kind of amino acid. So this gives you a probability of an amino acid in the position, and then that allows you to find like you can convert that to like what is not necessarily an energy, but just kind of still reflects probability of that amino acid in that context. So I opted to use this as opposed to something like FixBB, which is row-number based, just because I thought that it might struggle to find a suitable sequence if parts of the backbone were like odd. But like since then, reading about like how sequence design is done now with Rosetta, I saw like I think fast design, which also like relaxes the backbone a little bit. So I think there actually may be ways to do row-number based design that could be compatible with these kind of potentially imperfect backbones. This is just one example of the selected backbones with sequence designed and row-numbers built using fixed backbone. And I'm just showing it because it was interesting and I was able to kind of nestle into the binding site. It made some good charged interactions with the protein, but it also exhibits some issues like the fact that there are some clashes. It might be kind of hard to see like between this tryptophan and this methionine, which could potentially be fixed by relaxation, but obviously I don't know that. So I thought these designs were interesting, but there's definitely a lot of improvements that need to be made to the pipeline before we actually go out and test what it's generating. So now I have a really text-heavy slide. I'll try to like not take too long with it, but I want to talk about next steps because obviously this is still like really a work in progress. So I talked a little bit about coverage of native peptides, and I think that like I've argued that this RMSD cutoff like is potentially meaningful for defining coverage, but I think to actually really show that, I'd want to combine the seeds, like fuse them together and generate actual backbones, and sort of as a first step, just verify the quality, like make sure there's no like really like oddities in like the backbone geometry. But as a second step, I think it'd be much more informative to design sequence on those designed backbones and do kind of like a native sequence recovery test where I compare that design sequence to the sequence of the native peptide, which is a really hard test and I don't expect necessarily to do very well on, but I can also compare the sequences to that sequence that you can get designed on the native backbone. So that would be like maybe more comparable just because it's using the same kind of method. So if the method for designing sequence is imperfect, we'll like arrive at the same imperfect conclusion, but it'll still be evidence that like the backbone is similar enough that it's kind of seen as the same by these sequence design methods. Obviously going forward, we're also interested in showing how this could potentially be used to design new peptides. Right now we're working on doing this, focusing on known peptide binding sites. So I have a lot of the scores that I talked about, and I think it'd be interesting to compare those for like the designed peptides that I'm making and also the native peptides. So if our scores are like wildly different than native peptides, then there's definitely like something we're missing. But if they're comparable, at least with the resolution we have from those metrics, they're similar. I also want to design sequence using determine, and then build rotamers and compare. Actually, I guess I didn't fully complete this idea, but the idea would be to build rotamers and then look at the Rosetta energy and compare that to the native binder. It would be really interesting to also show that the peptide backbones that we've designed can sustain more than one sequence that has a reasonable energy. So we would basically try to design a sequence and then try to design another sequence with the constraint that it is within some number of mutations away from the first one. I think it would be promising to see if we could design lots of reliably low energy peptides that on the same backbone. And then finally, we can select some of our most promising designs and use molecular dynamic stability analysis, and also see if Rosetta Flex PepDoc ab initio could predict their structures. Obviously, I don't expect that for every individual example of a design thing, but I think on average, if either of these methods indicate that our design peptides are as good as native ones, then that would be pretty promising. This was basically this is stuff that I've been working on, but obviously it takes a village. Amy's super helpful with kind of framing ideas, and she's honestly very nice to allow me kind of to explore this sort of honestly bold idea early in grad school. Then Kat, who's just undergrad, who's now in grad school at CMU, who joined the project and really brought a lot of life to it. And he helped us also conceptualize and formalize some of these ideas and implement a lot of them in the code. And then finally, Gevorg is also really helpful for when I have technical issues or I want a second opinion on what I should do. So he's been a really big help with this too. Okay, that's all.
Awesome, Sebastian, excellent seminar. So yeah, let's open for questions for Sebastian.
Hi, Sebastian. Wonderful talk. So I had a bunch of different questions. The first one was, how do you account for entropy when you're dealing with such small peptides?
I mean, to be very frank, I don't. And I guess small peptides technically have less entropy than longer ones, right? But I guess you're pointing out the fact that these aren't necessarily binding in an ordered way. So there's a there's an entropic cost to them folding and then binding on the protein.
Yeah.
Yeah, so far I haven't thought about accounting for that. I think I've mainly thought about it as stabilizing the bound conformation as much as possible.
I see. Should I ask more questions or should I wait for other
Yes,
people?
definitely. Yeah, I might have better answers for those.
Okay, so this is more of an experimental question. So I had some experience of working with short peptides in my PhD, and what tends to happen for at least for the peptides that we had designed was, as you increase the concentration, they were meant to be helical, but when you increase the concentration, you ended up seeing a lot of cross beta interaction. So there was sort of a concentration limit that you just couldn't concentrate them beyond a certain point. So I just wanted to know if you had any thoughts on these kind of things.
Okay, so just to make sure I understand what you're saying that at the high enough concentration, the peptides bind to themselves in some way.
That's what we thought, at least. We did CD spectra, and that's what we observed. So we assumed that's what was happening.
No, I think that's pretty common. So if you're really certain that your peptide is a helix and that it's binding itself as a helix, you can kind of model that and try to incorporate that in your design and say I don't want a sequence that will stabilize this. I do want a sequence that will stabilize my conformation bound to a protein. But if you don't have any way to anticipate how they might be aggregating or binding each other, I don't know exactly how you would control for that or try to incorporate that in design. I think in general, if I guess I don't know at what concentration that was happening, but I guess if the concentration is high enough, then I would just consider the peptides to be not that good at binding if they were doing that instead of binding.
That's fair. And the third question I had was for your null seed models, when you randomize the positions, is there a way you avoid clashes? Because if you were to randomize any peptide near the surface, I would assume that it clashes with the protein.
Yes. Yeah. So if there was a clash with the protein, I would resample another conformation and new pose, and I continue until it was able to be positioned without any clashes.
Got it. Thanks.
Okay, I have a question, if you wouldn't mind. Thanks for the talk, Sebastian. Really interested in peptides here. And one thing I wondered about is if there are missed opportunities in the fragments or like the initial seeds. I know you assessed whether or not you have coverage of native peptides, but could you imagine it being beneficial to explore designs on top of native protein structures, then increasing your set size of, like, let's say even more, like let's say you're now stitching together many different design fragments. It just opens up lots of possibilities, and then you don't have to do a design step later. I guess I'm thinking about, this is philosophical, about not doing this backbone then design, but just having a whole bunch of design fragments. Do you know what I mean?
Okay, sorry. So I missed a little bit of it, but is the idea that like you can do a little bit of design on the fragments alone before you combine them?
Yeah, essentially that. Is that going to make your life better downstream if you maybe, or maybe, yeah, you're going to do design at the end anyway, doesn't matter.
Yeah, so I think that this is sort of related to this idea. I'll answer your question specifically, but we also thought about like scoring the individual seeds and then trying to sample paths that like had really high scores or included residues with really high scores. And we're still interested in that. It just kind of ended up being a problem of scale. It was actually really difficult to score all the seeds. And since then, our numbers have changed. But I think in terms of specifically designing sequence on the seeds, they vary in size, but some of them are rather small. And I think that just without the context of the things around it, there's potential that we wouldn't know exactly like what the proper sequence preferences of that seed would be. So for the rather small seeds,
you could design them in the context of the original PDB structure, even like the complex they were originally derived from, like if that would be possible, and maybe not inside the tight internals of a protein, but anyway, just
yes, right. There would be
brainstorms.
more information in that original context. I haven't quite thought about that. If there's some kind of way to capture that information that was flexible, so we weren't like assigning it sequence, but maybe giving it like a range of possible sequences, at least at each position, then maybe I could see that being incorporated. Yeah.
Hey, if I can ask one question, the picture you showed at some point, like you called, I think, hairy ball of the like seeds around the binding site.
Yeah.
There were some that were marked or rather colored as green, and some were colored differently, red, I think.
Yeah.
Is there any meaning behind that?
I kind of just like breezed past that, but the colors reflect the secondary structure of the, oh god, my laptop is like really not happy. It like is lagging. Let me see if I can just exit the slideshow, show it again. But anyways, the main point is that the colors correspond to structure. So we kind of expected there to be like clusters of different kinds of secondary structure seeds. So we colored them by that to sort of look for those trends, and we found them to some degree. It's a little bit harder to really pick out the trends for anything other than beta strands, but they're kind of in there if you look.
Right. Yeah. Right. There's a legend here. Right. I missed that. Sorry. So the other, like, sort of, I don't know, on the discussion side rather than a question, like when you resort to stitching multiple seeds right into a graph and then sample from that, I wonder if you only like use already so you make like those overlapping regions, right, as a way to switch from one seed to another, but then like say there were like two seeds like a little bit far apart from one from another, right? And were you looking maybe on, or what do you think for the on the whether like stitch them together, not through overlap, but just like empty region, fill it out or something like that? Like
when you're asking a
like
question,
not extend
oh, sorry.
to the end. Yeah, sorry, go ahead.
I was just gonna say, you can always look at Amy's face to know if you're asking like an interesting question that she's like quite fond of. So was there anything you wanted to add to that, or was it just kind of what you said?
No, that was just basically.
Okay. Yeah, so we've really kind of like, I guess the extreme example is that there's like two really good seeds that just, there's no like overlap to connect them. There's no way to link them by like drawing a path. And as of right now, like the way we've defined this just wouldn't be able to make a binder that has both of those things. But I think if we took this kind of like score then combine approach, then we could identify those. And if we wanted to, it'd be very easy, I think, to model in some kind of thing in between those two things. It obviously depends on how they're arranged, right? If they're like completely perpendicular, then maybe it'd be a little harder to like combine them. But if they're like joinable, we can definitely like link them together if that's all it takes.
Right, cool, yeah, thanks.
Thank you.
So I have a question. I think in this description, is this some subsampling of those like 2 million different seeds that you identified? I don't remember the precise numbers, but I remember it being a large number. Is it?
Right.
How are you actually doing all by all sort of search and comparison to identify locality and overlap and so on? Or are you doing something that's like more local in order to define? I'm wondering how you compose your graph, because that seems like a very expensive step. And then I had this other idea that's like not very fully formed at all, but I'll just say it. I kind of wonder if there's like some sort of trait space between like you're scanning through this, I don't know how you're iterating your seeds, but like you're scanning through proximal space when you go through a linear, as you may like go through the exterior of the protein. I just wonder if like as you do that, you maybe, if you kept some buffer or like some memory of the prior seeds that you put down and maybe scored fast, that you could say, oh, I want to do it to the n-1 set of seeds or something like that. So the first part is just like how computationally intensive and expensive is it to compose a graph where you just like slap on all these seeds, do some filtering maybe, and then compare them. And then is there a faster way to do that?
Okay, so I can answer the first question right off the bat. So my approach to this, of course, was just to like compare all possible overlapping windows. And like I built like a proximity grid where I was like, okay, things that are like more than this distance are not worth comparing. There's no way the RMSD will fall below the cutoff. There turned out to be like a limit to how fast you could do that, even if you tried to like break it up into batches, etc. So the undergrad I was working with was very like excited by this problem. And that's actually why we switched to using max atom deviation. So instead of comparing like all possible windows from all possible seeds, he computed the distance between every C alpha atom. And there's just like, there's less comparisons there. And it was a lot easier to then ask for like a set of multiple residues, whether for all of those, they all had like atom deviations below that cutoff. So that allowed us to break the problem down a little bit. But it is also very local in general, in that you're only looking for overlaps from things that are within some distance. So even though like there were a lot of seeds and that is a little slow, it wasn't as slow as you might imagine.
I see.
Does that answer that?
Yeah, I didn't appreciate that. Like as you place the seeds, you sort of bin them by their location, so you don't have to compute that per se again.
Right. And then your second question, I think I understood it, but I want some clarification, I guess. Are you talking about the step of sampling a path in the graph or a different step?
It was just like, could you use ordering on the protein that you're sampling as like a heuristic for, maybe these terms will seed to something that has closure? Like, it's not a fully formed thought. It was just like, are there heuristics that you can use in your sampling of the actual protein that you're targeting in terms of the order in which you may place seeds as a way of like very quickly to saying, okay, I should continue to place more seeds or not, but I'm not 100% sure that even makes sense. So
yeah, we just haven't thought about this kind of like live update approach where we like do a little bit of like generating seeds or combining them and then learn from that. I think it could be interesting, but I'd have to consider more like what exactly we'd be trying to do. Yeah, but that kind of idea of like learning as you're doing is pretty cool. Thanks for bringing it up.
So I have a question that's going to be also a little bit far out. Peptides are cool and all, but it seems like, I guess my question, why stop at a peptide? Could you keep doing the same thing and just keep growing this out into a full-blown globular protein?
Yeah, I mean, I definitely like have these kinds of like, oh, let's do this crazy thing ideas all the time, and I need to be kind of like harnessed if I want to get anything done. So I definitely spent a lot of time like imagining how fun that would be. And there's different ways to do it. I guess the most literal way is to design like a peptide and kind of treat it as like an interface, and then just like fix that and say, okay, now this is the new structure, build seeds around that, do it again. I think it could be done. I think you could also maybe just use a different method. Like, I think that what we're really doing here is prioritizing like complementarity to the surface of the protein you want to bind. But I think that like mini protein scaffolds already do a really good job of like prioritizing like foldability. So if there was like some way to like build a bridge, either like finding a way to like combine them, like just by searching for overlaps between the mini protein and that first layer, or building something between them, I think that could be a really useful approach. And that would kind of help with this conformational entropy problem that I have not been able to address yet.
Cool. Well, when you have really nice peptides bound to proteins and you want to turn that into a mini protein, we can help with that.
Cool.
I have a question. So how would this tool operate on like really deep grooves, thinking about like the GPCR typical pocket? Like that's peptide length that should be within your range, but I would think that the amount of interactions around in a helix might confound some of that stitching. I don't know.
I really wish I had now looked at the binding site on the GPCR. So you're saying on the like extracellular side, there's like a really deep binding groove?
Yeah, imagine just like a helix just jammed in a hole. How would
so I didn't quite do my homework. Some of those I think exist in the peptide protein complex database. I haven't quite like assessed them all because it's a little hard to search for them. There's no like label like that. But we still covered those. I think in some ways, I think that problem is a little bit easier because there's more constraints. It's like there's less space you can really put something. So either it fits and you can like see that based on the fact that it's making a lot of designable contacts, or it doesn't fit and like there's really no way to get it in there. That's kind of how I see it. I don't know if that's convincing, but yeah.
Okay, and maybe last one. I know peptides are cool and all, but what about small molecules? I feel like this could apply in principle the exact same way to stitching together some kind of, it's just that stitching step that I guess is a little bit hard, requires some medicinal chemistry knowledge, or not,
right.
Yeah, some chemistry knowledge.
But I just have to build the models, someone else synthesizes them, right?
Right, that's the advantage we have.
Yeah, it's a really cool idea. I think the DeGrado lab did a little bit of work with it recently came out. It's this paper where they introduced this like kind of structural unit Vandermeers. And they have a couple different like instances of them, but I think some of them are backbone dependent, and they kind of look at how like a rotamer can interact with like what do they call it, chemical group from, like generally they're taken from protein structures in the PDB, so like they're just like chemical groups you'd find on side chains, but I'm sure there's other databases that you could maybe use. And they use those to dock things. So they use those to dock small molecules, which have those chemical groups, into like potential protein binding sites. And it actually worked pretty well for them in their, at least the one example that they showed. But I think that you could, in principle, apply this to not only dock things, but also create new things. But yeah, you just have a new kind of stitching method. Okay, sounds like that's all. I really appreciate all the questions. Oh, AJ?
I think we have a couple of questions in the chat. Am I allowed to
Oh,
highlight
I have not even been
questions?
looking at that. Sorry, my bad. Yeah, that'd be really great if you could. Or I guess I can see them if I open the chat. Okay,
There's
yeah,
a question
whole
about
dialogue.
CASP, the amazing CASP results that just came out by Dylan, who's on a cell phone.
Oh, thanks,
There's
Gabor.
another one as well.
Okay, I'll try to march through it chronologically. Thank you for bringing this up, AJ. So yeah, this is definitely confined. Good call, Dylan. Motif score is sequence dependent. Right, confined can also be sequence dependent, as you can limit it to like rotamers of the sequence of the amino acid that is actually present at that position. Okay, so sorry, just a lot of reading. Dylan asks, sorry, on mobile, question. In the light of recent CASP results, and forgive the merely tangential relation here, but is there a straightforward and consensus objective that might be formulated in the same way CASP has, but for protein peptide interactions such as those presented today? Yeah, like the specifics have eluded me, but this idea of like framing this as like an end-to-end differentiable learning problem definitely like exists. And I think it'd be really cool if I could, you know, get this project done satisfactorily and maybe explore other ways of designing peptide protein interactions. So yeah, that's a really great question, Dylan. And if you want to chat more about that, I'd be happy to. I might butcher this name. Tongus asks, interesting idea. Have you tried to see if you can reproduce some known peptide binding structures in the PDB? Yes, I've tried to like cover them. Like, yeah, I don't know. I guess that's what part of the talk was about, but I haven't quite tried like redesigning them completely. So I think that would be kind of like a further step. Okay, is that all? Kind of sounds like it. Okay, thanks you guys. This was really great.
Awesome. Thanks again, Sebastian. Awesome seminar.